Hello everyone. In today's lecture, we will discuss DNA, deoxyribonucleic acid, that is the genetic material which is present within the cells, and also how it is organized. Before DNA was discovered, it was observed that organisms possess certain traits that appeared to be inherited by the offspring according to certain quantifiable and predictable set of rules. There were two main paradigms in biology at that time, and they were proposed by the great scientists Gregor Mendel and Charles Darwin prior to the discovery of DNA. Based on breeding experiments done with pea plants and also other systems, Mendel proposed principles of heredity that are known as Mendel's laws. In 1865-66, just to give you an idea about the timelines. And these findings were later promoted by later scientist Bateson, whose book cover is shown here. So Mendel's laws were basically dominance, that is, in offsprings having determinants of two varieties of a trait, only one, that is a dominant trait, is expressed. Now we know there are exceptions also. Segregation, organisms had two determinants of a trait, now known as alleles, and they segregated from each other during gametogenesis. And independent assortment, that is, the determinants of unrelated traits were inherited independently from each other. That is, they were independently assorted into gametes during meiosis. On the other hand, Darwin had proposed a theory of evolution by natural selection. According to this, variations were there in populations, but only the fittest survive, ultimately resulting in evolution. So it was in this backdrop that scientists wondered about the nature of the substance or the molecule that might be responsible for determining heritable traits. Based on the paradigms of inheritance and evolution understood at that time, it seemed reasonable that the genetic material should provide a rationale for the faithful inheritance of traits that were observed, and also it should provide a rationale for the differences in traits, or what is the cause of variations that now we know arise from mutations. As far as evolution is concerned, people wondered how do these selectable variations arise, and there should be a possible mechanism for this. At that time, by light microscopy, chromosomes were observed to appear in dividing cells as thread-like structures, and then these structures, they appeared to be segregated equally during cell division between the daughter cells. So these were very exciting observations, and people wondered whether they may have something to do with genetics, although it was not clear at that time. But chromosomes were also analyzed chemically, and it was found that chromosomes had DNA, deoxyribonucleic acid, as well as proteins as their components. So scientists were wondering, what is the genetic material? And of course, they knew that chromosomes had DNA and proteins, so these were two of the chief contenders. And people knew a lot about proteins, that they are made up of many building blocks, 20 amino acids, and that there are many different types of proteins. So it could have seemed at that time that organisms have so many different qualities and traits, and could there be some connection between proteins and the determination of those traits. And DNA, on the other hand, was also an important constituent of chromosomes, and it was known at the chemical level, it was analyzed, and it was known that DNA is composed of a sugar, phosphates, and 4 bases. So while people were wondering about this, in the 1940s, there were some interesting observations related to a pathogenic bacterium, Streptococcus pneumoniae. It was observed that there were 2 strains of this bacterium, smooth which was pathogenic, and rough, which was not. And these strains also, of course, as their name suggests, differed from each other based on their appearance, smooth versus rough. And when they were injected into mice, in case of the smooth strain, the mouse died, whereas in case of the rough strain, the mouse survived. And interestingly, it was observed also that when this R strain, which is non-pathogenic, is cultivated or mixed with either the inactivated heat-killed S strain or cell-free extracts from this pathogenic strain, then it was observed that when injected into the mice, now the rough strain could actually kill the mouse, and therefore it became pathogenic simply by being cultivated in the presence of the inactivated S strain or derivative cell extracts from it. So these were very interesting observations, and this phenomenon was referred to as transformation. Later on, scientist Oswald Avery and colleagues further tested the constituents of this smooth strain for the transforming potential. So they took different macromolecules, RNA, protein, DNA, etc., and then they incubated them with the R strain as per this experiment already described. And they found that only DNA from the S cells could transform the R cells to pathogenic form. So it was concluded that the transforming principle is DNA and not these other macromolecules. So what is DNA made up of? Basically, it consists of a sugar, bases, and a phosphate. So the sugar present in DNA is 2' deoxyribose. You can see the C2', it doesn't have a hydroxyl group, whereas a hydroxyl group at this position is present in RNA or ribonucleic acid that we will be discussing soon. So this sugar, deoxyribose, it has got 5 carbon atoms, as you can see, and it is a cyclized form of a pentose. There are 4 carbons in the ring, C1 to 4', and along with an oxygen atom, which is termed as O4', and the 5th carbon is not a member of the ring, but it is attached to the C4'. So a phosphate group is also part of DNA, and this is attached to the oxygen attached to the C5' carbon atom in a nucleotide, and in C3' in a dinucleotide or a polynucleotide, and that bond is referred to as the phosphodiester bond. Bases present in DNA are heterocyclic nitrogen containing molecules. Okay, there are 2 types, pyrimidines and purines. So pyrimidine has got a 6-member aromatic ring with 2 nitrogen atoms at positions 1 and 3, and it is one which gets attached to the sugar, and it's got 4 carbon atoms and various side chains or groups. The purines have got 2 fused 5 and 6 atom rings, and the positions 1, 3, 7 and 9 are nitrogens and the rest are carbons, and it is the 9th position that gets attached to the sugar. The main bases present in DNA are cytosine and thymine are the pyrimidines, and guanine and adenine are the purines. So the chemical constituents of DNA were known, and it had been found that the ratio of the amounts of A to T and G to C are close to unity in DNA by Chargaff in 1952. Several groups of scientists were interested in solving the structure of DNA particularly after the experiments of Avery demonstrating that it was likely to be the genetic material. These included Wilkins et al., Franklin and Gosling, who were trying to solve its structure by X-ray diffraction, and Watson and Crick, who were trying to deduce the structure based on the information that was available to them. 3 papers were published in the same issue of Nature in 1953, reporting their conclusions regarding the structure of DNA. Watson and Crick hypothesized, based on published experimental data, including Chargaff's, and the stereochemical arguments which were based on model building that they were engaged in, that the DNA consists of 2 helical chains which were coiled around the same axis. These are right-handed helices, but they run in opposite directions, that is, they are anti-parallel. They propose that the chain consists of phosphodiester groups joining the beta-D deoxyribofuranose residues with 3' 5' linkages. The bases were on the inside and the phosphates were on the outside according to this model, which was different from an earlier model proposed by Linus Pauling, and also Watson and Crick had come up with that model, which was not published by them though. They also proposed that in this current or new model proposed by them, the residues recur every 3.4 angstroms in the z direction, and there are 10 residues for 1 full turn after 34 angstroms. And the distance of the phosphate from the axis is 10 angstrom. This implied that it should have a diameter of 20 angstroms, and that the planes of the bases are perpendicular to the axis. Purines paired with pyrimidines of the opposite strand, and the base pairing was rather specific, that is, A would only pair with T and G with C. Wilkins et al. Had an X-ray photograph of DNA and they interpreted it, and they also proposed that the helix would have a pitch of 34 angstroms, a diameter of 20 angstroms with 10 nucleotides in 1 turn, and also that the nitrogenous bases would be in the central region of the helix. Franklin and Gosling had obtained a very high quality X-ray diffraction image, famously known as photo 51 nowadays, which was the label that they had given to that photograph, of DNA from calf thymus. And this was actually a critical evidence in determining the structure of DNA. This is the structure of the more hydrated B form of DNA. And this pattern, which they observed in the X-ray diffraction image, which you can also find by looking up this paper as well as this other article, it was consistent with a helical conformation. And they also proposed that there are 10 residues per turn, and the helical structure had a diameter of 20 angstroms. The phosphates were on the outside, sugar and bases turn inward. And there were 2 coaxial molecules with a cylindrical repeat unit of 34 angstroms. So very similar conclusions by the 3 groups, and this gave rise to what we now understand as the double helix Watson and Crick model of DNA structure of bDNA. So according to the model, DNA is a double helix, a beautiful helix shown here. This is an artist's depiction, but it's aimed to be accurate in terms of dimensions. And this helix, as you can see, it resembles a spiral ladder. Okay, with the bases would be the rungs of the ladder in this case. So DNA is a double helix with the sugar phosphate backbone on the outside and the base is on the inside, and the 2 strands, they run in opposite directions, that is, they are anti-parallel, and also they are complementary with the specific base pairing which happens via the hydrogen bonds. So let's try to understand the structure of the double helix in more detail, starting with the components. So one of the main components is sugar, 2'-deoxy ribose. I already mentioned that in case of RNA, at this position there is a hydroxyl group. The phosphate is attached to the C5' of one sugar, and the base is attached to this oxygen attached to this C1' of the ring, and it forms an N-glycosidic bond. The N1 of a pyrimidine and N9 of the purines are connected to the sugar at the C1' position. There is also phosphate group I already mentioned, which is attached to the C5'. These are the structures of the bases present in the DNA, and what you can observe is that they have certain differences. They have, for example, cytosine has got an amino group, so has guanine, and thymine and adenine. They have distinct groups attached to them, which accounts for the differences among them. The basic building block or repeating unit of DNA is a nucleotide. It consists of a base, a sugar, and a phosphate. The nucleotide AMP, adenosine monophosphate, is shown here with one phosphate and the deoxyribose sugar and the base adenine. The phosphate is attached at the C5' position, whereas the base is attached via the NO glycosidic bond at C1' of the sugar. A DNA polymerase can use a nucleotide triphosphates as precursors and can add a second nucleotide via a phosphodiester bond at the C3' of the first connected to the C5' of the next nucleotide. And thus a dinucleotide as well as a polynucleotide would have a free 5' phosphate and a free 3' hydroxyl end. For the growth of the chain, the hydroxyl group at the 3' position on the terminal nucleotide attacks the linkage between the alpha and the beta phosphate groups of the incoming NTP. One nucleotide gets added and a pyrophosphate ion is released, resulting in the formation of the trinucleotide as shown in this example. This slide shows base pairing found within DNA. So each base has got unique locations of hydrogen bond acceptors and donors based on these different groups which are attached to them. For example, in case of adenine, this amino group is there at C6 position, and it can serve as a donor for 2 hydrogen bonds. And also in this case, the lone pairs of electrons at the nitrogens 1, 3 and 7 impart a partial negative charge, so they can act as hydrogen bond acceptors. And likewise, guanine has got this CO group and an amino group at 2, and this oxygen here is an hydrogen bond acceptor. The amino would be a hydrogen bond donor. So the base pairing is specific. It is based on the potential of these groups to form the hydrogen bonds and their orientations relative to each other. So the base pairing that occurs in DNA is shown here. You can see that cytosine and guanine, they are bonded by 3 hydrogen bonds in this configuration, and adenine and thymine are bonded by 2 hydrogen bonds. This diagram shows the specific base pairing scheme which is present in the double helix. So in this diagram you can note that the 2 strands, they are anti-parallel. Okay, 5' to 3'. And 5' to 3' in this direction. So this scheme, it suggests a mode for replication or copying of the genetic information. The bases, as you know, they are complementary. So it was famously stated in the paper by Watson and Crick that it hasn't escaped our notice that the specific pairing that we have postulated immediately suggests a possible copying mechanism for the genetic material. When you have one strand and you know the sequence of it, the other strand, as per rule, has to be complementary. So this was thought to be very exciting by everyone in the field. This animation shows the three-dimensional structure of DNA. Note the phosphate groups, these large yellow spheres on the outside, and the bases containing these navy blue atoms on the inside. Also observe the major groove, which is broader, and the minor groove, which is narrower. So to summarize what I've said so far, DNA is a polymer consisting of 4 nucleotides, bases linked to phosphorylated sugars, and the nitrogenous bases, they are purines and pyrimidines. Purines, adenine and guanine, the pyrimidines, cytosine and thymine. There's a 5-carbon sugar which is part of the DNA molecule building block. It's 2' deoxyribose, and there is a phosphate group attached to it, to the C3' and 5' of successive sugars forming this phosphodiester linkage. A DNA is a double helix. It's got 2 anti-parallel strands which are twisted around each other. Purines pair with pyrimidines of the other strand via hydrogen bonds, A with T via 2 hydrogen bonds and G with C via 3 hydrogen bonds. And in the B form of DNA, the rise per base pair is 3.4 angstroms. There is 1 full turn every 34 angstroms, and there are 10 bases per turn. And the diameter of the helix is 2 nanometers. So here's a space filling model of 1.5 turns of a DNA double helix, and you can see that each turn, that is from here to here, is made up of 10 nucleotide pairs, and the center to center distance between adjacent pairs is 3.4 angstroms. The coiling of the 2 strands around each other, it creates 2 grooves. Okay, the wider one is referred to as the major groove, and the narrower one is referred to as the minor groove. So the bases in the major groove, they are more accessible and probably can be more readily recognized by DNA binding proteins. This slide illustrates the semi-conservative replication of DNA. So the 2 parental strands, they would separate, and each would serve as a template for the synthesis of the new daughter strand shown here in gray by complementary base pairing. And this model, in fact, we know this to be true, and this was also confirmed experimentally later on by Messelson using DNA labeled with the heavy isotope of nitrogen N15 and coupled with density gradient centrifugation using cesium chloride density gradients. It was observed that intermediate density DNA could be observed after shifting from the heavy to the light isotope of nitrogen, and in one round of DNA replication, and this was consistent with semi-conservative mode of replication. So there was a very nice boost to this model. In this animation, made and provided by Drew Berry and Etsuko Uno, we can see how the DNA is organized within the cell. In the cell, the DNA, whose backbone is negatively charged, associates with basic histone proteins that form an octamer around which the DNA is wrapped. The histone octamer has got 2 each of 4 histones, H2A, H2B, H3 and H4, and there is also a linker that is associated with histone H1. The histones are shown here in blue. This unit of chromatin organization is referred to as a nucleosome. It forms a 11 nanometer diameter fiber, which is further folded to form a 30 nanometer diameter fiber. Note that the histone tails are exposed to the outside. These can be modified. This thread is further folded to form what we are familiar with as the folded chromosome structure seen in metaphase. And the organization of this is just beginning to be understood. It perhaps happens by formation of loops and their further folding in association with non-histone proteins as well. We have discussed that base pairing is specific. However, there are no restrictions on the type of sequence for the successive or adjacent bases next to each other on one strand. This can be seen here as the output of a sequencing reaction. You can have, different variations of DNA sequence. So there can be any combination of successive bases forming the sequence, and this sequence of bases actually corresponds to the genetic information. Here is a list of some of the key references for this lecture that will be useful to read up. Stay tuned for the next lecture on the decoding of genetic information.